All right, it is my pleasure today to introduce Dr. Mary Torregrossa, who is an associate professor at the University of Pittsburgh. She'll be telling us a little bit about a paper that her and her trainee Sierra Stringfield recently published in the journal Psychopharmacology. It's called Intravenous Self-Administration of Delta-9 THC in Adolescent Rats Produces Long-Lasting Alterations in Behavior and Receptor Protein Expression. Dr. Torregrossa, could you tell us a little bit about the main finding of this paper?
Yes, so the goal of this paper was to investigate whether adolescent self-administration of THC, which is the primary psychoactive component of cannabis, if it produced long-term consequences in adulthood on cognitive function. And so we assessed this in rats, and we used a test of working memory in adulthood called the delayed match to sample task. And so initially rats were trained to press a lever to receive intravenous infusions of THC. And so that was one of the first findings of the paper, or first, I guess, things to establish in the paper, was whether adolescent rats would actually self-administer THC intravenously. Because historically, it's been very difficult to get rodents to voluntarily take significant quantities of cannabinoids in general. We had previously published studies showing that we could get a synthetic cannabinoid, which is more potent than THC, to be self-administered by adolescent rats. And in general, we found that our adolescents were much more willing to self-administer cannabinoids than adults overall. So we do think that some of the prior research that had difficulty, part of the issue was actually just the use of adults relative to what we see with adolescents. So my postdoc, Sierra Stringfield, established a paradigm for THC self-administration. It involved actually slowly escalating doses over time in adolescents. And then she was able to maintain them on a unit dose that produced what we ultimately term like moderate THC intake and higher levels of THC intake, allowing us to determine basically a dose effect curve of amount of THC in adolescence that leads to different cognitive outcomes in adulthood. We also compared males and females. And so after they self-administered during adolescence, and we got them to these kind of two different dose ranges, they just sat for a few weeks and we started training them on this task. And in the task, we have a chamber that has 5 nose poke ports. They can be individually controlled, so we can illuminate one. The rat learns to poke its nose in the illuminated hole and ignore the others in order to get a food reward. And ultimately, we train them to the point where they make a response on the illuminated port. Then there is a delay of where all the ports are extinguished and nothing happens. And then 3 of the ports light up, and they have to remember which was the one that they had originally sampled and respond again in that port in order to get rewarded. And if the delay is very short, so if the light goes on in the center hole, they respond, it goes off for half a second, it comes back on, it's very easy for them to respond again in the correct center hole and get, correct responses that are around 90–95%. But as that delay increases and they have to wait longer and longer before the choice option occurs, their behavior becomes worse and worse, and they get to a point where they're responding just at chance, they're just guessing. So this is assessment of how good their working memory is. The longer they can remember the correct response, the better their working memory. And what was interesting is that the animals that had adolescent THC exposure, self-administered in adolescence, compared to a group that just self-administered saline, in the adult males, they actually had better working memory performance than the saline controls. And this was particularly true of the group that self-administered actually the highest dose of THC in adolescence. And actually the higher their intake correlated with the better working memory performance in adulthood. And so this is somewhat unexpected based on human literature, where people generally think that use of cannabis is bad for working memory and cognitive function in general. However, our females were a little bit of a different story. So the females didn't really show a significant difference in their working memory performance overall. But if you looked at the correlation between how much THC they took in adolescence and their performance, then we saw a negative correlation. So the more they took, the worse they did. So it's the opposite of the males, suggesting that there are sex differences in the way adolescent exposure affects adult performance. And then we also looked at changes in the expression of proteins regulating working memory function in prefrontal cortex and other regions. And we did find this long-lasting reduction in CB1 receptors. This is the cannabis 1 receptor that responds to endogenous cannabinoids and THC, was downregulated in a few brain regions in adulthood, regardless of whether they're male or female. And additional proteins regulating GABAergic and glutamatergic function were also downregulated, suggesting that there is long-term consequences in the cortical function that might underlie these changes in performance. So those were the primary findings of that paper.
Oh, very interesting. So if I heard you correctly, you're saying that THC, when self-administered by adolescents, actually can improve cognitive performance or working memory, at least in male rats, although it has detrimental effects apparently in female rats. Is that correct?
Yes, that's correct.
Fantastic, very interesting. And could you tell us a little bit about the sort of backstory of this paper? How did you decide to do this study?
Yes, so there's a couple of different reasons. So one was that actually locally here at the University of Pittsburgh, there were some ongoing studies, both in human adolescents and then in some non-human primates on campus, looking at either cannabis users versus never users in this longitudinal cohort of people they have been studying here at Pitt for a while. And then in adolescent monkeys, then looking at working memory function as they aged. And so there were actually some interesting findings suggesting that the cannabis users were not showing deficits in working memory that they expected based on kind of some older human literature, whereas the monkeys were showing some more of a slowing in their maturation of working memory throughout development with cannabis. And so we decided that it'd be good to just get a rat model where we could actually do self-administration so that the animal can control how much it takes and take the amount that it wants, as opposed to these other studies where the experimenter just picks a dose and gives it to them with, daily, and the rodent or the monkey doesn't have any control of the situation, which could be stressful and add a confound. And so what we do find is that, rodents, when they self-administer cannabinoids, the total doses they're getting are much lower than a lot of the prior rodent studies looking at cannabis or THC effects on learning and memory, suggesting that, if you go up to high doses, then you probably do see some longer term deficits, but that might not be relevant to the doses that most humans use, especially those who use socially. There are other issues. A lot of the human studies is often that people are using other substances. And so that's another advantage of the animal models. We can isolate the effects of THC, whereas the human subjects that may be cannabis users, but they're also often nicotine users or smokers or vapors, or they are alcohol drinkers and other things. And so it gets to be a lot more complicated. And so that was a lot of the motivation behind it, is really just being much more systematic about understanding how self-administered doses of THC would ultimately affect adult behavior. And so I think that makes it one of the sort of clearer studies looking at it. And then we've since there's been support. Well, we've done we've done a synthetic cannabinoid too and had similar results. And then we have colleagues at other universities who've used slightly different models, but have also seen signs of adult improvement in various types of cognitive tasks with the adolescent exposure.
Wow, really interesting, especially to see these effects across species. Do you have any sense of whether the sex differences that you found also extend across species?
Yeah, no, that's an interesting question. And it may be different in other species. The human literature, unfortunately, is very unclear in terms of just historically, we haven't been very good at delineating effects that might be specific to males or females in general. And often the sample sizes are not large enough. In the non-human primate study, it's a little less clear. It looked like potentially the females were a little bit more protected. So there might be something different. But again, the paradigm was different because it wasn't self-administration of the drug.
Really interesting. And so could you tell us if there's anything else that you really think is kind of outstanding as the important next step? Like what do we not know that we really need to know next?
Well, for my lab, we're really interested in the mechanism of the improvement, right? I mean, I think it's really interesting that there's something that can be, started in adolescence that leads to this, change in brain function that might actually improve cognition. And so figuring out how that works, like what could do that, might be useful for a lot of other purposes where we might want to improve cognition. And so we're actively just kind of investigating that microcircuit in the prefrontal cortex where the CB1 receptor and GABA and glutamate are signaling, and where we know we see these changes in protein expression, and trying to figure out exactly what cell types are showing these changes, and so how that affects the function of basically excitatory inhibitory balance of GABA and glutamate. And then we're also doing other studies, recording neural activity during the working memory task to see if we could see differences in the way the neurons encode or how they behave when the animal is performing the task after adolescent THC self-administration.
Very cool. Looking forward to hearing more about those findings. As often occurs, I think some of the most important findings, such as the ones you have in this paper here, often just lead to us understanding how little we actually know about the mechanisms, and shows us that we have far more questions to answer yet, even though we've made great progress step by step. Great. And so can you also tell me, I know you are an author that has published quite a few times in the journal Psychopharmacology. Would you like to say anything about your experience publishing with the journal, or with the editorial process, or the kind of science that you think fits well there, and that kind of stuff?
Yeah, so Psychopharmacology has been a journal prominent in my career ever since I was a graduate student. I did my PhD in the lab of Jim Woods at the University of Michigan, and he published with Psychopharm frequently. Everybody did, and has just a strong history of publishing very strong behavioral pharmacology findings, and findings with high scientific rigor. I know that throughout my career, the editors such as Klaus Mischek and Trevor Robbins and others basically took such an interest in their papers that they handled, and would personally give advice on how to address a reviewer's comment, or a better way to do your statistical analysis, and to make sure that everything was of the highest scientific rigor. So I actually find that papers published in Psychopharmacology to be of very high quality and much more reliable or trustworthy than you might find in some high impact journals where they let some of those things slide sometimes. And so they've always published, really interesting, and foundational findings in the field that have led to many exciting directions that people have gone often in their careers.
Absolutely. I think it's been a crucial journal for the field of behavioral pharmacology long before we got here. So great. Well, this has been a great talk. Thank you so much, Dr. Torregrossa, for the time, and look forward to seeing your next paper, perhaps in psychopharmacology.
Thanks, Steve. It was nice talking to you.